Hello and welcome to Molecular Views. I'm your guide, Dr. Brandy Cross, and today we'll be exploring the cannabis plant with an introduction to cannabis biochemistry. This is one of the videos in a series of educational videos on popular biochemistry, so please subscribe for more. If you'd like early access and bonus clips, please think about becoming a Patreon at the PotLab PhD. Today, we will rediscover cannabis as a plant. We'll talk about why it was outlawed, and we will talk about cannabinoids. In fact, two very popular ones. We'll discuss how cannabinoids have been used as medicine and how they act on the human body. And now the legal red writing at the bottom. This program is for educational purposes only and does not serve as medical advice. Please consult with your physician before starting any kind of herbal treatment. And now let's begin. So, our first factual statement is cannabis is illegal due to racism. We say this because there's no conclusive scientific, medical, or even metaphysical evidence that cannabis causes harm. In fact, the old studies that used to be cited showing cannabis causes brain cell death were actually studies done on brain cancer cells. And when they tried to redo these experiments from these old models, they found that cannabis does kill brain cancer cells, but it actually does not kill healthy brain cells, which is an interesting fact. When we studied pregnant mothers in Jamaica, we found that the users that used a lot of cannabis throughout their pregnancy, in fact, gave birth to children with slightly above normal intelligence, according to IQ tests. So the only thing we can look to is the historical evidence for racist legislation, at least in the United States. And whatever the United States does, the world follows. In the United States, cannabis is closely associated with enslaved peoples. This is because daga seeds came with African slaves, and Native Americans from south of the border migrated to the United States with cannabis because in Mexico, the Spanish had enslaved the indigenous people to grow hemp for their sailcloth. Hemp and marijuana are the same plant. And as we move towards legalization, we seem to forget that cannabis was legal up until the 1930s, when Henry Anslinger made a series of racist and sexist arguments to create the first Bureau of Narcotics Enforcement. This legacy continues into modern times as we fight to decriminalize and legalize cannabis all over the United States. So let's talk about the truths. Cannabis is a plant. And here's my friend watering his cannabis plant. Because if it's a plant, it needs water. So let's simplify this. Cannabis is not toxic or poisonous. It has no spikes, thorns, or spines. And it loves the sun, a little bit of water, and there's tons of plain honest truths we can learn from it. So just to prove once more that it is a plant, it's angiosperm. That means that it makes a seed and pollen. That means that the plants are sexed. There's a female version of a plant and a male version of a plant. Intersex plants or hermaphrodites are often a result of female plants getting some kind of light stress. Cannabis plants are the order Rosales. This is the same order as, yes, roses. The cannabis plant is in the family Cannabaceae with hops. Yes, hops that's used to brew beer. The genus is actually named Cannabis, and there aren't real species due to the domestication of the plant. You see, much like the domesticated cat, cannabis has been domesticated so long that these species seem to intersect and don't have a true genomic difference. Cannabis is often called a weed because it can tolerate a range of mild equatorial climates. Naturally, cannabis germinates in the spring as an annual, and it grows throughout the summer in its vegetative stage. And then come the fall, with the dryness and a decrease in light, it begins to bloom. You can see here the difference between a male flower, which looks more like a pod, and a female flower, which begins to grow hairs. We can recapitulate these growing conditions indoors or in a laboratory. The female flowers are compound flowers, much like basil or lavender. These female flowers are the flowers sought after when we talk about buds. The female flowers additionally develop high amounts of trichomes to prevent pests, mildew, and mold from these large, thick compound flowers. This protects their seeds. Cannabis plants make cannabinoids to protect their seeds because this keeps lots of bugs and pests away. To a lower extent, the male flowers will produce some trichomes to protect the pollen. However, since so much pollen is made, there are less trichomes on the male flowers. When we talk about medical cannabis grows, we're often talking about all female plants because seeds are often not sought after unless you're doing genetics work. The female plants are often called mothers, and they are cloned off by their branches to make more and give a constant supply of blooming female cannabis plants. So let's look deeper into these trichomes and how these little structures make cannabinoids. Hemp or marijuana? That seems always to be the question. And the true answer is it's all just cannabis. All cannabis plants make cannabinoids. These trichomes, or mushroom-shaped glands, are the site at which all cannabinoids are made. Cannabinoids exist in a default carboxylic acid-bound state within the trichome secretory cavity. Historically, it was thought that cannabinoids existed within these plants in their decarboxylated state. Remember that guy that told you his dog got loose and ate his live marijuana plant and got high? Well, he was lying. Because in the living state and in the fresh state, and in fact, most of the time, THCA is how THC is stored in the cannabis plant. THCA is a different molecule and has its own medical applications. And in another video called Cannabis in the Raw, which is coming up, you will be able to see how THCA is used in medicine. However, we're going to look at the historical scientific issue. How did it happen that we thought that THC and CBD were stored in the plant as such? Well, chromatography was a process always used to separate and identify things in mixtures. And in the 1950s, gas chromatography was used to identify volatile compounds such as cannabinoids. This process uses increasing heat to identify molecular parts or compounds in a mixture. It was used widely in chemistry for many, many applications, except it identified compounds via degradation, which means everything that went through it was somewhat degraded before it was identified. By the 1970s, we had developed HPLC, which is high pressure liquid chromatography. It involves the use of two different solutions and pressure to separate compounds in a mixture, thus leading to no degradation of natural compounds. And this is how we found that 99.9% of the potential THC in cannabis is stored as THCA. Looking inside the trichome, we find two cell types, the stipe cell here and the disc cell in orange, which lead to a secretory cavity, which is in gray. Over 95% of the cannabinoids that are manufactured are made in the trichomes. There are two types of trichomes, tall and short, and cannabinoids are made here from phenol glucosides secreted from the stipe cell. The disc cell releases CBGA that it makes from the phenol glucoside into the secretory cavity. And depending on what type of plant you have, a cyclization into THCA or CBDA will occur. CBDA and THCA are stored as such because the carboxylic acid group seems to stabilize the compound within the secretory cavity. In order to create THC or CBD, you'll need a substantial heat source to undergo a reaction called decarboxylation. So, to talk about THC and decarboxylation, we'll need to discuss a little bit about THCA. THCA is a different compound altogether from THC because before it's decarboxylated, it has a different molar mass and a completely different molecular formula. In my next video, Cannabis in the Raw, we'll discuss further all the great things about THCA, but for the purposes of this video, we will note that THCA is a precursor to THC. So when we talk about decarboxylation, we're specifically talking about the removal of this carboxylic acid group in the square here to form THC or CBD. And this is how it happens in THC. And for hemp plants, we're going to talk about CBDA being decarboxylated into CBD. And in my next video, Cannabis in the Raw, we'll talk specifically about CBDA more. But for the purposes of this video, it's worthy to note that CBDA is a precursor to CBD. In a nearly identical chemical reaction, CBDA's carboxylic acid group can be removed via heat or UV irradiation to form CBD. CBD will not get you high, which has caused a great interest in using CBD instead of THC for various medical reasons. CBD, however, has limited medical use despite its exponential popularity. And in my video, Cannabis in the Raw, you'll see why that may be. As more cannabinoids are purified and tested, we find even better molecules for certain conditions. So it's always good to stay updated on the latest research. Okay, doc, but tell me, what does cannabis do in our bodies? Well, that's a complex answer there. You see, the human body makes its own cannabinoids, which we refer to as endocannabinoids. The cannabinoids from cannabis we call exocannabinoids. And in our bodies, these work together to get us high, relieve our pain, or just put us in a better mood. So let's start off in the most important place in the body, the brain. Cannabis affects all parts of the three-part synapse made by the pre and post synaptic terminus and the astrocytes. Cannabinoids are retrograde neurotransmitters which affect the postsynaptic neurons to affect presynaptic neurons. That's working backwards. Additionally, THC and CBD inhibit the clearance of endocannabinoids, again, which our own bodies make, and act on microglial cells and astrocytes, which are the immune system of the brain. This is very promising from those suffering from neural inflammatory diseases, depression, anxiety, ADHD, some forms of autism, and more. Coupled with the interaction with serotonin receptors, we see interesting effects to mood and anxiety with the addition of exocannabinoids. So let's look at the molecular mechanisms of cannabinoids at the cellular level. I'm sure you've heard the rumors that cannabis cures cancer and thought it was all crazy. But for years, cell biologists have been trying to study exactly why that brain cancer cell experiment went awry. And they want to know what is killing the cancer cells. So through tons and tons of work and years of study, we've actually discovered several pathways by which cannabinoids regulate the cell cycle. When we say cell cycle, we mean the reproduction of cells. When the proliferation of cells is out of hand, it forms a tumor. Those tumors can be benign and proliferative, or they can start migrating, which is called malignant. And that is when we say you have cancer. See, it's different just to have a tumor and to have cancer. Tumors are often found in the adult body and removed with great success. However, migrating tumor cells are always a problem. And cannabinoids affect tons of pathways, which essentially shut down both the proliferation and the migration of diseased cells, including cancer cells. So, this is a molecular mechanism of tons of work. We're talking decades of work from hundreds of researchers. So, every time you see one of these little pathway images, just know this is from tons of data over the years. So let's summarize the cannabinoid pathway by its receptor activation and consequences. What we want to remember here is that THC binds preferentially to the CB1 receptors, found mostly in the brain, and CBD tends to preferentially bind to CB2 receptors, found mostly in the immune system. These molecular pathways lead to outcomes in which there's whole body regulation and modulation. Additionally, the interaction of these receptors and these molecules with molecular pores, such as TRPV1, changes the calcium dynamics in all cells. This often results in the apoptosis or cell death of diseased cells. In addition, we have the modulation of serotonin in the brain, helping with different mental health disease states. Cannabis affects every part of the human body by intervening at the cellular level to modulate the cell cycle and cellular signaling. Thus, using medical cannabis is something you should proceed with caution and always discuss with your physician. Here we'll discuss different ways of utilizing medical cannabis. So, ingesting cannabis responsibly takes some knowledge of the types of dosing. The first and most popular way to ingest cannabis is by smoking. And this is actually very useful for people suffering from mental health issues such as depression and severe anxiety, as well as people with ADHD and pain. Yes, pain. This is because the onset of smoked cannabis, the effects are felt within 5 minutes. Because the pathway from the lungs to the blood is very short, THC and CBD are basically absorbed immediately, and you feel effects. You can imagine if you were in a lot of pain, this would be your favorite way to ingest cannabis, whether that pain is emotional or physical. The second most popular way are edibles, and these are for people who are on moderate to high dose cannabinoids. Heating or baking cannabis is going to decarboxylate it, and then it can be absorbed in the stomach as THC or CBD. However, edibles, again, are usually at a higher dose, and thus their time and duration to onset is going to be a little different. It could take up to 2 hours to feel the effects of an edible. So please proceed with caution, because while you may not feel it an hour after you eat it, in 2 hours you may feel it very strongly. Edibles are highly recommended for chronic pain and for people who are suffering from the effects of chemotherapy. Additionally, edibles have been used to combat anorexia and bulimia. Infused oils are a third way, but they're much like an edible, again with a effect of 2 to 4 hours. This is when you infuse oils with cannabinoids by soaking the plant in your favorite oil and then straining it off. Many cannabinoids are typically found in an edibles type mixture. Now, if we look at concentrates, these waxes are often smoked for recreational purposes, inducing a psychedelic effect. However, concentrates have been made in the edible form for treatment of cancer. Yes, treatment of cancer. The diagrams I showed you earlier of the effects on the cancer pathway by cannabis require up to 1,000 milligrams of cannabinoids per day. Preferentially, it seems THC concentrates. Now, these concentrates last 1 to 3 hours when they're smoked, but 4 to 6 hours if they're ingested orally. This means this kind of treatment is usually the last resort before very, very strong medications are used. Now, the last way we're going to talk about ingesting cannabis is through topicals, which are really popular with senior citizens. These skin ointments, lotions, balms, and even patches can be used at the site of pain without inducing a high, even if there's THC in the mixture. So, these site directed cannabinoid therapies have been super popular in legalized states, again, in senior homes, and it's a way to stop the potential abuse and addiction to any cannabinoids if that's an issue. In addition to cannabinoids, cannabis, like many other plants, makes terpenes. And here are certain of the terpenes that cannabis makes. As you can see, they all have medical uses and are found in other plants. So if you're interested in this list, just take a pause here and you can see what plants you can also find these terpenes in. Many of these terpenes are found in the essential oils of very common plants. So cannabis dose versus medical issues. As a cannabis counselor, I get asked a lot what dose and what kind of cannabis to use. Well, in this introductory video, I'm going to introduce you to our dosing chart. All adolescent patients are recommended a low dose therapy, so that's just a note there. Child and adolescent epilepsy, ADHD, ADD, mild anorexia, bulimia, and metabolic diseases often fall in the low dose spectrum. This is 1 to 10 milligrams of cannabinoids per day. In the moderate dose spectrum, we have many other disorders, including adult epilepsy, immune disorder, severe ED, depression and anxiety, autism, pain inflammation, muscular ataxia, and PTSD. The reason these all fall into the moderate dose range is these are adult doses. In our high dose category, you'll notice there's very few candidates. Cancer and lymphomas take up most of the space in the high dose group because we've seen results with and without chemotherapy. It seems that cannabinoids have a tendency to protect the body when it's going under chemotherapy. Additionally, lupus and autoimmune disease patients undergoing chemotherapy have sought out high dose cannabis therapies to increase their quality of life. So I like to always include a little harm reduction. If you're a first timer, you can use this checklist. Just pause and print. Make sure that you take these precautions the first times you use cannabis. Always have a medical expert, experienced user, or psychedelic counselor available for your first dose. You can do this physically or digitally. If taken orally, use the lowest recommended dose and wait 90 minutes for the effects, especially if using tinctures, oils, or other edibles. Having a day's supply of food and water and even nutritional beverages is highly recommended as your appetite may suddenly increase. This happens whether using THC or CBD. Create a safe space in your home with comfort, activities, and items which make you happy. This will help curb any anxiety that's felt. And remember, always consult with your physician before starting a new herbal therapy. And this concludes this introduction to cannabis biochemistry. Please like and subscribe so you don't miss our next video, Cannabis in the Raw, exploring THCA and CBDA. If you're new here, please consider subscribing for more videos. And if you love this content, please consider joining my Patreon. The information is in the description. And with that, this is Dr. Cross giving you another molecular view.